Hello students. In previous two lectures, I told you about the structure of DNA, its components also, nucleotide structure, nucleoside structure, nitrogen base structure, how a DNA helix is formed, means how a double bond is formed between adenine and guanine, and how a triple bond is formed between guanine and cytosine, right? We already discussed about this. And now here, the previous lecture we finished our lecture at that point, right? The structured DNA become very clear. Now here today we are going to start from this silent feature of double helical structure of DNA, which was given by Watson and Crick, right? And according to the Shergaff rules, Shergaff's equilibrium rule, Shergaff tells that adenine is always equal to thymine and guanine is always equal to cytosine, or we can say also purine is always equal to pyrimidine, right? Here, the silent feature of double helical structure of DNA are as follows. What is given here? First, it is made up of two polynucleotide chain where the backbone constitutes by sugar phosphate, right? And the base project inside. Base project inside, right? Base means it looks like, okay, DNA structure looks like a stair, right? Ladder like structure is there. A twisted ladder, we can say here twisted ladder. Twisted ladder means the sticks in between these two strands, right? They are made up of nitrogen base and their hydrogen bonds, right? And the strand, it is made up of sugar and phosphate. That is given here consist by sugar, phosphate and the base project inside. Okay, the two chain have anti-parallel polarity. Already we know one is 3' 5', another one is 5' 3'. It means if one chain has the polarity of 5' 3', this 5 and 3 they indicates the number of carbon. Means they indicate the phosphoric acid attached with the 5th carbon of pentose sugar, and that side phosphoric acid of another nucleotide is attached with the 3rd carbon of the pentose sugar, and the other means other is just opposite, it is 3' to 5', that like structure, right? This one antipole parallelism. The base in two strands are paired together, hydrogen bonds, okay, forming base pair. They form the base pair, right? Adenine forms two hydrogen bond with thymine. Already we know how they form, right? With thymine from opposite strand and vice versa. Similarly, guanine is bonded with cytosine with 3 hydrogen bonds. So here 3 hydrogen bonds are formed. As a result, always the purine comes opposite to pyrimidine. Purine comes opposite to pyrimidine. This generates approximately uniform distance between the two strands of helix, right? Who is responsible for the uniform distance between two helix, right? Who is responsible? That adenine and purine. They comes always opposite to one another. That's why, and Shergaff's equilibrium law is also responsible for this one. This one means what? Uniform distance between the two strands of DNA. Okay. Now the two chains are coiled in right hand fashion. Fourth one is given, two chains, they are coiled in right hand fashion, right? Right hand fashion means what? This one, right? You can right handed fashion. Right handed fashion means what? What do you mean by right handed fashion? It is anti-clockwise or clockwise. We can say this right hand fashion means what? It is clockwise fashion. The pitch of the helix is 3.4 nanometer. Already we discussed about 3.4 nanometer, and or we can say it is 34 angstrom. It is that it depends upon the unit which we use here. So 3.4 nanometer. A nanometer is the 1 billionth of a meter, that is 10 power 9 meter, and angstrom is what? It is 10 power 10 meter. And there are roughly 10 base pair in each turn, right? In each turn, 10 base pair. That means what? In each means between two base pair, what is the distance? It is 0.34 nanometer, or we can say 3.4 angstrom, right? This structure is a DNA structure. Here you can understand. Look here, double standard distance. This one is the first strand. This 5 dash, you know here 5 dash, 3 dash we can say, because here look here, this 3 dash means here 3 dash is free, and this side what, this 5 fifth carbon, this one is the fifth carbon, and fifth carbon attached with the phosphoric acid, that is the phosphate moiety, and that's it, that is hydroxyl moiety. But in case of second, what happens, that just opposite, here phosphate moiety is present at that place, but here phosphate moiety is present at this side, just opposite sides, and this side it is hydroxyl moiety is there on that third. Okay, I already tell you here in previous class that when we see the structure of pentose sugar, we understand what is the moiety, means what is the direction. You can see here, that is the fifth carbon, third carbon. In a single, if we see, this is a pentose sugar, this one is pentose sugar. In pentose sugar, you can see here, this is the fifth carbon of pentose sugar, and this is the third carbon of pentose sugar, right? When we 1, 2, 3, 4, and 5 pentose sugar. So what is the moiety? 5 dash, 3 dash, 5 dash, 3 dash. A single nucleotide, we can understand what is the direction of the nucleotide. But in this case, what happened? In here, what you see? Here you see this is the fifth carbon, 1, 2, 3, 4, and 5, right? You understand here, this is the third, means we can say that is the 5 dash, 3 dash, just opposite. So through a single nucleotide, we understand what is the direction of this strand, right? This is the backbone. Backbone means that is phosphate plus sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. That is called phosphate sugar backbone. That side also sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate. That is the sugar phosphate bond. And these are the nucleotide. These are the nitrogen base, right? Toward the inner side, right? Inner side means adenine, thymine, double hydrogen bond, thymine, adenine, double hydrogen bond. But guanine, cytosine, this triple hydro bond, right? Already I tell you how they form, right? When in previous lecture you can see easily, right? Because I draw in previous chapter and previous lecture, I draw the molecular structure of this double DNA helical structure. Okay. Now another structure is given here. Simply when we see this is the structure of DNA, coiled structure. We can say here a coiled or twisted ladder-like structure, right? These are base pairs, and this is the sugar phosphate backbone. This one, right? This sugar phosphate backbone. This one is right. Means it is made up of sugar, then phosphate, then sugar, then phosphate, like this one, right? And this side it is a nitrogen base. If it is purine, that means what? This one is pyrimidine, right? Adenine, thymine, cytosine, guanine, it is randomly arranged. This is the structure of our DNA helix. Another one thing I tell you here, the distance between these two, right? It is 20 angstrom, or we can say 2 nanometer, right? And one is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. From this side to this side. This one is 3.4 nanometer, or we can say 34 angstrom, right? Distance between these two, right? These two, it is 3.4 angstrom, or we can say 0.34 nanometer. Okay. Consequently, the distance between the base pair in the helix is approximately 0.34 nanometer. As I already tell you, 0.34 nanometer. It is also, we can say this one, that is a 3.4 angstrom. It is depend upon what unit we take. The plane of one base pair stacks over the other in double helix. This is additional. The H bond confers the stability of the helical structure, because here they one upon another is there, it becomes a stable structure, right? So these are some some characteristics or properties of DNA helix is given. We can say a silent feature. Compare the structure of purine and pyrimidine. Okay, already we compare this structure of purine and pyrimidine. We know purine means it is double cyclic structure, but it is single cyclic structure. Hexagonal, but it is hexagonal plus pentagonal, right? Can you find out why the distance between the two polynucleotide chain in DNA remain almost constant? Yes. Why the means the distance between two? Because purine always come up just if one side is purine, another side is pyrimidine, right? The purine always come in complementary to the pyrimidine, or pyrimidine one purine is there. That means only purine pyrimidine is there. No purine or pyrimidine is there, right? That's why a uniform distance is present in between two strands, right? The proposition of the double helix structure for DNA and its simplicity in explaining the genetic implication becomes revolutionary, right? Very soon, Francis Crick, that is the scientist, proposed the central dogma in molecular biology. Central dogma. Dogma means what? Central idea of molecular biology. Molecular biology means what? How a life continues on the earth. We understand here. Central dogma of biology means what? Here, Watson and Crick, Watson and Crick, they give the model, right? But this Crick, only Crick, Francis Crick, he gives the central dogma of molecular biology. So, central dogma of molecular biology means what? DNA. What happens in our body? DNA means it forms RNA. And RNA it forms protein. This is the central dogma, and it is one-way direction. Look, DNA through replication, it forms DNA. This process is called replication or duplication, right? And DNA forms RNA. This process is called transcription, right? And RNA forms protein. This process is called translation. So DNA forms DNA, that is replication or duplication. DNA forms RNA, transcription. RNA forms protein, that is translation. And this is the total, means we can say this is the life. This one is life. What is metabolic activity? For metabolic DNA forms RNA because our body needs enzyme. Without enzyme, no function is going on. No metabolic activity is going on. So, enzyme is what? Protein. How they form protein and who is responsible for protein, for the formation of protein? We know very well. Our ribosomes, they are the factory of protein. In ribosomes, messenger RNA, they have code for the formation of specific protein. Transfer RNA, they are also involved. Right? So first, what happens? First, DNA has to form RNA for a specific protein, right? By using their code. So this process is called transcription. And second, when a messenger RNA is formed, now it comes out of the nucleoplasm and come into the cytoplasm, and on ribosome it forms the protein. All the metabolic activity is going on, so that, right? Okay, when we see the cell division, or we can say formation of gametes or reproduction, or as well for continuity of species, what is here? DNA replication. DNA forms. In S phase, number of DNA content will be double. Number of chromosome is equal, but the number of DNA content means number of chromatin thread, it becomes 4n. That is replication. Means whole world or biological activities, or we say metabolic activities, or continuity of a species or life, means it is totally depend upon these three terms: DNA to RNA, RNA to protein. First thing. So this one is one-way direction. That is called central dogma. Second, in cases of some virus means which virus I am talking here? This virus is retrovirus. Retrovirus means retro comes from reverse transcriptase. Look here. Sometimes what happens here? DNA. What happens here? DNA. Now it forms RNA. Already we know RNA. And RNA forms protein. This is the central dogma, right? But sometimes what happens, if it is a RNA virus, as for example our HIV, or this time it becomes coronavirus, right? RNA, because virus is RNA forms DNA, right? When RNA forms DNA, So this transcription is called reverse transcription. Reverse transcription because here what happens? RNA just opposite, it is just opposite the central dogma. It is just opposite transcription. What is transcription? Transcription means formation of messenger RNA. That is transcription through DNA. But when RNA forms DNA, it is also transcription, but it is reverse transcription because its direction just opposite of transcription, right? So this one is called reverse transcription, and then the new DNA which is formed in host DNA, right? It forms another RNA. They may be viral RNA or some RNA. They form the capsid proteins for the newly virus. So this process is called reverse transcriptase. Cryptase, right? So reverse transcript. This one is a reverse transcript, right? And this one is transcriptase. Now I think you understand, right? Difference between the transcription and reverse transcription. Okay. Transcription means what? When DNA forms RNA or messenger RNA is formed by DNA, then it is called transcription. And when DNA is formed by RNA, in case of virus, when RNA forms DNA, right, then it is called reverse transcription, right? And these viruses are called retrovirus. Re for reverse, pro is transcriptase. Okay. So this one is another, but it is cyclic. Now this here is cyclic central dogma. We can say cycle because DNA, RNA, then RNA, DNA. A cycle is formed, so we can say here cyclic central dogma, right? And major means what? In our body, not in virus. This reverse transcriptase is present only in some viruses. That is only RNA virus, we can say, but not in other organisms, right? Cellular organisms means only one thing is there. DNA forms RNA, RNA forms the protein. Because viruses are, we understand the viruses have no protoplasm, that means they are dead, they are non-living, right? So if we consider the cellular organisms, we have only one central dogma, that is DNA to RNA, RNA to protein, a linear central dogma. We have two types of central dogma, linear central dogma and circular central dogma. Circular central dogma is limited up to retroviruses, right? RNA viruses, but this linear DNA is applicable for all the cellular organisms. We can say all the living organisms. Okay. Here. Very soon, Francis Crick proposed the central dogma in the molecular biology, which states that the genetic information flows from DNA to RNA, RNA to protein, right? So when DNA forms RNA, it is called transcription. Here it is given. You can see here, right? This DNA forms RNA, messenger RNA. This process is called transcription. And when messenger RNA, because it has code, it forms protein, then it is called translation, right? And when DNA forms DNA, that is called replication, DNA replication. And this whole linear, right? This is called central dogma of biology, molecular biology. Or central dogma means what? Central idea. This is the central idea or central dogma of molecular biology, which is stated by Francis Crick, not Watson. Do you understand? Okay. In some viruses, already I told you, in some viruses, flow of information is in reverse direction, and that is from RNA to DNA. Can you suggest a simple name of this process? So we can say yes, it is the reverse transcriptase. Reverse transcriptase. This process name is reverse transcriptase. An example for retroviruses. Right? These are the examples. Now, packaging of DNA. How long? Because we know human DNA is approximately 2.2 meter long, and this 2.2 meter long DNA is placed in a very simple, very small cell, which we can very hardly we can see there, right? How it is arranged or packed inside the cell. This is packaging of DNA helix. Now it is called nucleosome. Here a nucleosome structure is given, right? So taken a distance between two consecutive base pair as 0.3 nanometer, or we can say 0.3 into 10.9 meter. If the length of DNA double helix is typical mammalian cell is calculated, right? Calculated, how can we calculate it? Because we know in human diploid cell 6.6 into 10 power 9 base pairs are there. We have to multiply this one with the distance between two nucleotides, and that is 0.34 into 10 power minus 9 base pair. Now we get it is near about to 2.2 meter, near approximately. It comes out to be approximately 2.2 meters. Okay, a length that is far greater than the dimension of a typical nucleus, not in cell, but it is inside the nucleus. Nucleus means we cannot see this naked eyes. Approximately it is 10 power minus 6 meter, right? How is such a long polymer packaged inside the nucleus, right? We know a very long thread, when it is reeled, it is placed a very small reel structure, right? You know thread, and it is we can say it warm, right? So it is what happens. When it is reeling over a pipe, then what happens in very long, means more than 1 kilometer thread, it comes into a small structure. That also here packing happens. If the length of E. coli DNA is 1.36 millimeter, can you calculate the number of base pair in each coli? Yes, you can say we can calculate, because we have to divide it with that means what, 0.34 into 10 power minus 9 meter, right? When we divide this one, we can get the number of total nucleotides, right? Base pairs, number of nucleotide or base pair, we can say. If the length of E. coli DNA is 1.36 millimeter, can you calculate the number of base pair into E. coli? Yes, you can say we can calculate, right? Because we have to divide this one 1.36 millimeter. The millimeter means what? 1.36. We can say 1.36 multiplied by 10 power minus 3 meter upon, second one is our 0.34 into 10 power minus 9 meter. Then we get, if you calculate this one, then you get the answer. It is your duty, right? You can calculate this one. E. coli means it is already given, right? 4.6 into 10 power minus 6 we can get, because when we 4.6 is somewhere, right? It is 4. We can get 4.6 multiplied by 10 power minus 6, right? Meter. That is, this is the number of 10 power 6, not minus 6, 10 power 6 base pair, right? 10 power 6 base pair. In prokaryotes such as E. coli, though they do not have a defined nucleus, because prokaryotes, they have no nuclear membrane, so we can say they have no defined nucleus, right? The DNA is not scattered throughout the cell. That means what? That means not the DNA is scattered throughout the cell, but they are misplaced in a specific location. So DNA being negatively charged, we know it is a negative charge PO4, right? Phosphoric acid is there, negative charge, each held with some proteins, and these proteins that have positive charge. DNA is always negative, so all the proteins which are related with DNA, they are positive charged, right? In a region termed as nucleoid, and this region is called a nucleoid. The DNA in nucleoid is organized in large loops held by proteins. DNA in nucleoid means what? False nucleus, because it has no membrane, right? No nuclear membrane. Nucleoid is organized in a large loops which is held by proteins. Which proteins? Positive proteins. Okay. Now, first you see this one, this structure, and then this is the structure of nucleosome. Nucleosome means it contains DNA helix, second H1 protein, histone octamer, right? H1 protein and histone octamer is there, right? Here, you know, this one is H1 histone, and this octamer means what? It is made up of 8 monomer of histone protein, right? Look here, this wraps 1, 2, not complete 2 circle is there, only 1.75 circle is formed here, right? And in the second structure, here we can see here, this structure, a bead-like experience. These beads are nucleosomes, right? These beads are nucleosomes. Electron microscopic picture, beads on string. Now what is given here? In eukaryotes, this organization is much more complex. There is a set of positively charged basic proteins, and these proteins are called histone. Histone protein is absent in prokaryotes, right? A protein acquires charge depending upon the abundance of amino acids, residue with charge size chain, right? Histone are rich in the basic amino acid residues. Basic amino acid means which one? Lysine and arginine. Actually, we have three basic amino acids are there. Only two acidic amino acids are there. Already we discussed about in the biomolecules, right? So, two acidic and three basic. Basic means lysine, arginine and histidine, right? And in case of histone protein, it is lysine and arginine is present in more number. That's why this protein is base. This protein is positive charge and base, right? This one. Both the amino acids residue carry positive charge in their side chains. Histones are because they have extra amino group. These means of amino acids, they have extra amino group, NH group. That's why they are positive in charge. Histones are organized to form a unit of 8 molecules. That is called, because it is made up of 8 molecules, so it is called octamer, right? The negatively charged DNA is wrapped around the positively charged histone octamer to form a structure that is called a nucleosome, right? Negatively charged DNA wrapped around positively charged histone octamer. Now a structure is formed, and this structure is called nucleosome, right? A typical nucleosome contains 200 base pair. How much? 200 base pair of DNA helix. A single now we can understand, if 200 base pairs are present in a typical nucleosome, that means we can calculate how many nucleosomes are there. Your answer is yes. We can calculate. We have to divide this 200 base pair with the total number of base pair. Then we understand how many beads are present, right? Or how many nucleosomes are present. Nucleosomes constitute the repeating unit of structure. Nucleosome, they constitutes the repeating unit of structure in the nucleus, and that is called chromatin, right? Thread like stained colored, right, bodies seen in the nucleus. That's why they are called as nuclein, right? Protein. Because the nucleosome in chromatin are seen as beads on string, right? Chromatin means what? Chromatin, a protein, because at that time he did not understand what is this, right? Misher did not understand what is this, so he said them nuclein, right? And chromatin means what? Chrome means what? Colored bodies, a colored protein, because this chromatin, they are stained, right? So they are named as chromatin. Okay, so chromatin are seen as beaded on string, as is seen here. You can see here string structure when viewed under the electron microscope. Theoretically, how many such beads nucleosome do you imagine are present in a mammalian cell? In a mammalian cell, we have to divide 200 base pair, right? That means the total base pair, number of base pair is divided by 200 base pair. Then we get the total number of beads, right, in a specific mammal, right? The bead on string structure is in chromatin is packaged to form chromatin fibers that are further coiled and condensed at metaphase stage, right? That means they are super coiled structure, right? Then they form solenoid. Ultimately, they form chromosomes. Stage of cell death to form chromosomes. The packaging of chromatin at higher level additional set requires additional set of proteins that collectively are referred to as non-histone chromosomal protein, NHC protein. Non-histone, they are not histone proteins, but they are additional proteins, other proteins, right, which are responsible for the formation of nucleosomes, or we can say chromosome, or the packaging of nucleotides, right. These are called non-histone chromosomal proteins, right. In a typical nucleus, some region of the chromatin are loosely packed. Loosely packed, okay, in previous lecture we understand that linkage. So linkage means what? When they are means this linked, they are linked group. If they are linked very closely, that is called densely packed. If they are linked, their distance is too much high, means it is more, then it is called loosely packed, right? And we know very well, loosely packed means their mutation can be easily happens. Their crossing over also can easily happens there, and also because they can these two strands, they can easily separate with one another. That means what? These are active. Active means they are active for replication, for transcription, right? So they are called as, because they are not for replication. Mostly they are responsible. These are the part responsible for the formation of messenger RNA, and the process is called transcription. So we can say they are transcriptionally active part of DNA. So this transcriptionally active part of DNA is different in different cell. It is depend upon the type of cell. It is depend upon the function of cell, right? As for example, that if enzyme is formed, a specific enzyme is found, like insulin is found. Insulin structure is different. Insulin amino acid sequence is different. So a different part of in our body, all these cells, they have the same. We have same type of chromosomes. 46 chromosomes are there, but their active, transcriptionally active part is different. That's why different cell forms different enzymes or different proteins. Do you understand this one? That is given, right? Here, in a typical nucleus, some region of chromatin, that is DNA, are loosely packed and stain light and are referred as euchromatin, right? The chromatin that is more densely packed and stained and dark, and they are called as heterochromatin, right? Euchromatin is said to be transcriptionally active chromatins, right? Because they are loosely packed. So they are transcriptionally active chromatin, means this euchromatin is responsible for the formation of messenger RNA, or we can say euchromatin is responsible for transcription. Okay. Whereas heterochromatin is inactive. Heterochromatin, which is densely packed, it is inactive. They are right. They are present, but they are not active. This euchromatin and heterochromatin is different means different in different type of cell. Different part of DNA is euchromatin and heterochromatin. Okay. Mostly euchromatin is toward the central region, and heterochromatin is toward the peripheral region in nucleus. Now, 5.2, it is given search of genetic material. Search of genetic material. Genetic material means which one is genetic material. How the search, right? Here three means this is given here. Three stories given, we can say, for the searching of this DNA, right? Even though the discovery of nuclein by Miescher and the proposition for principle of inheritance by Mendel were almost at the same time. They are at the same time, right? 1869, near about the same time, we can say, right? But that the DNA active genetic material took long to be discovered and proven. Very long, means near about, in the last lecture, we understood it is near about more than 90 years, right? By 1926, the quest to determine the mechanism of genetic inheritance had reached the molecular level. Previous discoveries by Gregor Mendel, he said them factors, Walter Sutton, Thomas Hunt Morgan, right, and numerous other scientists had narrowed the search to the chromosomes located in the nucleus of the most cells. But the question of what molecule was actually the genetic material, which one is actually responsible for the inheritance of characters or properties, right, has not been answered. They discovered, but they did not. They have no idea who transmit the genetic information from one generation to another generation. Okay. Now the first scientist, he is Frederick Griff. In 1928, Frederick Griff is in a series of experiments with Streptococcus pneumoniae. Streptococcus pneumoniae, right? Responsible for the pneumonia. Bacterium responsible for pneumonia. You understand Streptococcus means a chain-like structure is formed. This chain is made up of coccus. Coccus means you understand is circular structure, right? Pneumonia. Bacterium responsible for the pneumonia witnessed a miraculous transformation. Miraculous, wonderful transformation in the bacteria, right? During the course of his experiment, a living organism, that is bacteria, has changed in physical form. How? Because a rough bacteria convert into a smooth bacteria, right? Non-infectious bacteria converts into a infectious bacteria, right? That is why it is called miraculous transformation. When Streptococcus pneumoniae, that is pneumococcus, bacteria are grown on a culture plate, right? Some produce smooth, shiny colonies. Smooth, shiny colonies, why they form smooth, shiny colonies? Because they have a mucus coat. And this mucus is made up of polysaccharides, right? You will read here, are grown on culture plate, right? Some smooth, shiny colonies, that is referred as far as smooth, shiny S. While other produce rough colonies because they have no mucus, so they are dry and rough, right? They are produced as rough colonies are for rough, right? This is because the S-strain bacteria have a mucus polysaccharide coat, while all strain they does not. Mice infected with, look here, this smooth, it is infectious, but this rough, it is non-infectious, right? Mice infected with S-strain, that is virulent, here said, die from the pneumonia infection. But mice infected with R strain does not develop any symptom of pneumonia, right? Do you understand here? We understand that S strain means it is virulent, it is responsible for the pneumonia, but R is not responsible for the pneumonia. Here it is given, when S strain is injected into mice, this mice die. And when R strain injected into mice survive, right? Then what happens? Griffith was able to kill bacteria by heating them. Now he took out bacteria from the dead mice. Means he took some sample, means blood from this mice, right? In blood they have bacteria. So he boiled them. When he boiled them, what happens? These bacteria get dead. He observed that the heat killed S-strain bacteria injected into mice did not kill them. Then this dead bacteria he injected into the healthy mice, what happens? No infections. Then he understand that means these S-strain bacteria, they are dead. Okay. Now what happens? When he S-strain heat killed, inject into mice survive. But when he put this bacteria, S-strain killed bacteria and this rough, both in a single test tube, right? What happens? That is S-strain heat killed plus R-strain live. We understand this. If we inject this S-strain heat killed, mice survive. If we inject R-strain, mice survive. But when we enter this mixture, S-strain and R-strain, then what happens? When it is injected into the mice die. And when he took out some blood from this mice, he not found this R-strain, but he found S-strain bacteria, S-strain live bacteria. Then he understand from where S already they are dead, but R-strain they are converted into S-strain. Then he said them transformation, and he astonished how R is converted into S, right? This process called transformation. Injected the mixture of the heat killed S and R bacteria, the mice died. Moreover, he recovered living S bacteria from the dead mice. This S bacteria means these R bacteria. We have only live R bacteria is there, no S live bacteria is there. But this R bacteria is miraculous, or we can say it is converted into S bacteria. Okay. He concluded that R-strain bacteria had somehow been transformed by the heat-killed S-strain bacteria. Some transforming principle is transferred from the heat-killed S-strain, right, and had enabled R-strain to synthesize a smooth polysaccharide coat and become virulent. That means today we understand that some part of this DNA means heat-killed S-strain bacteria DNA is entered into the R-strain bacteria, and this RNA-strain bacteria means it also forms the smooth, that is polysaccharides, right, and this is responsible for the infection, right. But at that time he did not understand how this must be due to the transfer of genetic material. However, the biochemical nature of genetic material was not defined from his experiment. They understand some material is there which is responsible for transformation of R bacteria into S-strain bacteria, but in this experiment they did not get which material is responsible. Now second, three scientists, they came and they start another experiment. Biochemical characterization of transforming principle. Same experiment, that is Streptococcus pneumoniae experiment is there, but here scientists have prior to the work of Oswald Avery, Colin MacLeod and McLean McCarthy. Year 1933 to 44, the genetic material was thought to be a protein. At that time, they think that genetic material is a protein, right? So they work to determine the biochemical nature of the transforming principle in Griffith's experiment. They purified first they separate the biochemicals, that is protein, DNA, RNA, etc. And then they use digestive enzymes. We can say hydrolase enzymes, right? First they use protease enzyme. So what happens? Protein is digested, means no protein is there. That means what? Transformation occurs. That means protein is not responsible for transformation. Then they use RNAs. This RNA, it is dissolved, but transformation occurs. That means RNA is also not responsible. They use protein and RNA both at the same time. Transformation occurs. That means these two are not responsible. But when they use DNAs, no transformation is there. No transformation. Then they understand only DNA is responsible for transformation. That means what? DNA is the genetic material. Okay, here from the heat-killed S cells to see which one could transform live R into the S cell, they discovered that DNA alone from S bacteria caused R bacteria to become transformed, right? They also discovered that protein digesting enzyme, that is proteases, and RNA digestive enzyme, RNases, did not affect transformation. So the transforming substance was not a protein or a DNA. Digestion with DNAs, this is the enzyme, ASE means enzyme, right, did inhibit the transformation, that suggesting that the DNA caused the transformation. They concluded that DNA is the hereditary material, but not all biologists were conceived at that time, right? Can you think of any difference between DNAs and DNAs? We can say yes. Why? What you understand? DNAs means it is the plural of DNA, more than one DNA, and DNA is ASE. This ASE means enzyme. ASE means enzyme. Maltase, sucrase, protease, lipase, amylase, RNase, DNase. That is what? ASE means what? Enzyme. So the DNA means enzyme. The genetic material is DNA. No. Hershey and Case in 1952, they proved that DNA is only responsible for transcription, or that DNA is only responsible for the transformation of genetic materials, right? The unequivocal proof that DNA is the genetic material came from the experiment of Alfred Hershey and Martha Case, two scientists, in 1952, right? They worked with viruses that infect bacteria, and that is called bacteriophage virus, right? The bacteriophage virus attached to the bacteria and its genetic material then enter into the bacterial cell. This is a common process we know already. The bacteria cell treats the viral genetic materials as if it was its own and subsequently manufactures more virus particles, right? This is the working pattern of retroviruses, right? Hershey and Case worked to discover whether it was protein or DNA from the virus that enter into the bacteria. Who enter into the bacteria? Then they used radioisotopes, right? Radioactive. They grew some viruses on the medium that contained radioactive phosphorus and some other on medium that contained radioactive sulfur. We know very well this phosphorus, it is present in nucleic acid, right? Only, but phosphorus is not present in protein, and sulfur never present in nucleic acid. It is present always in protein, right? So they use this one. Viruses grown in the presence of radioactive phosphorus contains radioactive DNA, right? And similarly, the virus grown on radioactive sulfur, they contains radioactive protein but not radioactive DNA, because DNA does not sulfur, right? Radioactive phages were allowed to attach to E. coli bacteria. Then the infection proceeded. The viral coat were removed from the bacteria by agitating them in a blender. The virus particles were separated from the bacteria by spinning them in a centrifuge. Through centrifugation, this one is right. They separate these particles. Bacteria which was infected with viruses that had radioactive DNA were radioactive, right? That had radioactive DNA were radioactive, indicating that DNA was the material that passed into the cell, bacterial cell, right? And bacteria there that were infected with viruses, right, that had radioactive proteins, right, were not radioactive. That means this protein cannot enter into the bacteria. That the protein did not enter into the bacteria from the virus. DNA is therefore the genetic material that is passed from the virus to the bacteria. Bacteriophage. Here 3 steps are there, and these 3 steps is infection, second blending, attachment, third centrifugation. Through centrifugation we get separate them, right? And you understand here radioactive S35 label protein capsule. S35 means what? Here red color protein is there. So this red color protein is radioactive protein, right? But DNA, it is in black color, means it is not a radioactive. Okay. When you see in this month after infection, what happens? This DNA enters into the bacterial cell, but this protein coat it remains outside the bacteria. In third, what happens here? Centrifugation. When we know radioactive S35 detect in the cell. This is a normal bacteria, means no radioactive sulfur is detected here. That means what? Radioactive S35 detect in the supranatant, detect into the viral protein capsid, we can say in the capsid. But here what happens? It is radioactive P32 labeled DNA is there. So this red color you can see here. This DNA is the radioactive DNA. It enters into the bacteria. Now when through centrifugation, when we separate this DNA, we understand this DNA, it is not a normal DNA, but they are radioactive DNA, and that's proof that DNA enters into the bacterial cell and it is the responsible for genetic inheritance. And supranatant, they have no radioactive, means protein, they have no phosphorus. So you understand there is a 3 steps: infection, blending and centrifugation. Hershey case experiment. Properties of genetic material. Now DNA versus RNA. What are the properties which are essential for genetic materials? We have 4 properties which is discussed here. From the foregoing discussing, it is clear that debate between protein versus DNA as the genetic material was unequivocally resolved. From the Hershey cage experiment, it becomes an established fact that it is DNA that acts as genetic material. However, it subsequently becomes clear that in some viruses, RNA is the genetic material. In RNA viruses, they have no DNA. In RNA, they have genetic material is RNA, right? For example, tobacco mosaic virus, plant viruses, they are RNA virus, we can say they. So, tobacco mosaic virus or QB bacteriophage virus, they are RNA viruses and they have genetic material is RNA, right? Answer to some of the questions such as why DNA is the predominant genetic material. So, why DNA is the dominant genetic materials in all the living organisms except some viruses only, right? Whereas RNA performs dynamic functions of messenger adapter has to be found, messenger adapter structural, right, has to be found from the difference between the chemical structures of the two nucleic acid molecules, right? Here, can you recall the two chemical differences between DNA and RNA? Already we read the structure of DNA and RNA. The chemical differences means what here? One difference, because in RNA, you can say here, look, DNA means two difference. That side RNA. What are the two difference? First difference, DNA contains thymine, RNA contains uracil. First difference between pyrimidines. Okay. Second difference, pentose sugar. It contains deoxyribose and it contains only ribose. So we can say here C5H10O4, but here this one is O4. Here C5H10O5. That means what? Here second carbon, it has only H group, but here second carbon it has OH group. That is the main for stability, right? These are the two chemical differences between DNA and RNA. A molecule that can act as genetic material must fulfill the following criteria. How 4 criteria are given here. First, it's called replication, we can say, right? Second, stability. Third means what? Mutation. And fourth means what? Replication. It should be able, first one, it should be able to generate its replica. It should be able to generate its replica means it can replicate itself. That process is called replication. Second, it should be stable chemically and structurally. It should be stable to store the information, genetic information. Third, that is called stability. Third, it should provide the scope for slow change, that is mutation, for evolution, that are required for evolution, because we know evolution is continuous process, right? So first mutation, that is for mutation. It should be able to express itself in the form of Mendelian character, that is called expression, right? What are the 4 basic criteria here? Replication, stability, mutation and expression. You understand what are the 4? It is replication, second one is stability, third one is mutation and fourth one is expression. That are the 4 basic criteria for a genetic material. Now, if one examine each requirement one by one, okay, now we are going to examine this one. Because of the role of base pairing and complementarity, both the nucleic acid DNA and RNA have the ability to direct their duplication. They have ability, right? The other molecules means both have this replication power, right? The other molecules in the living system, such as protein, they fail to fulfill this criteria, right? Means this duplication or replication, they can form their replica. It is who can form their replica? Only DNA and RNA both can form, but protein they cannot form, right? So first criteria is present in both RNA and DNA, that is replication. Second, the genetic material should be stable enough not to change with the different stage of the life cycle, that is age or with the change of physiology of the organism or environment, right? stability as one of the properties of the genetic material was very evident in Griffith's experiment. We understand because bacteria dead, but its DNA molecules, its DNA is not dead, right? It is active. When it come into the another cell, it starts its process expression. Okay. Itself, that heat which killed the bacteria at least did not destroy some of the properties of genetic material. Properties of genetic material means properties of genetic material means what? DNA. This shows, this now can easily be explained in the light of DNA that the two strands being complementary, it separated by heating, come together when appropriate conditions are provided. When we heat these two DNA, they get separate because they have hydrogen bond. But when we cool them, they come and bind once more, right? And that's why that means in Griffith's experiment, this bacteria get dead. At that time, these strands are get separated. But when they cool, then what happens? They once more form the double helical structure, right? Further, second thing, second carbon contains OH group, hydroxyl group present at every nucleotide in RNA, right? Each a reactive group. Hydroxyl group is a reactive group and makes RNA labial and easily degradable, right? That's why retroviruses, they are easily mutate. RNA is also known to be catalytic. Ribozyme is there, enzymatic activity is there. Hence, it is also reactive. Therefore, DNA chemically is less reactive and structurally more stable. Chemically less reactive means what? Because second carbon contains only H group. Second, structurally more stable means what? It contains double helical structure, right? When compared to RNA. Therefore, among the two nucleic acids, the DNA is the better genetic material. When we consider the stability, then we understand that DNA is more stable, so it is the better one. In fact, the presence of thymine at the place of uracil. Now, third one is there. Because uracil, what is the difference between thymine and uracil? Because thymine, it contains methyl group, more stable. But uracil, it contains only H group, that is less stable, right? So, here, if the presence of thymine at the place of uracil also confirms additional stability to DNA. Detailed discussion about this requires understanding of the process of repair in DNA, and you will be studying these processes in higher classes. Okay, in your bachelor degree or in master degree, you can read this one. One thing is there because that is thymine, it contains methyl group, so it is less reactive. But uracil structure are same. Only one difference is there, methyl and hydrogen. But uracil, it contains only hydrogen, which can be easily separated, right? Which can be easily replaced, right? That's why RNA is stable, but the DNA, which contains thymine, is more stable. RNA is not stable, right? RNA is less stable. Here, both DNA and RNA are able to mutate. In fact, RNA being unstable, mutate at a faster rate, right? Consequently, viruses have RNA genome and having shorter life span, mutate and evolve faster. RNA genome in some viruses, right? It is responsible for mutation and more evolved means they can evolve faster also. Both DNA, okay, here, RNA can directly code for the synthesis of protein. RNA is independent. RNA can code for the synthesis, right? But DNA for the formation of protein, it is totally dependent upon the RNA. First, it has to transcript a messenger RNA, then a protein is formed. So, the RNA can directly code for the synthesis of protein, hence can easily express the characters. DNA, however, is dependent on RNA for synthesis of protein, right? DNA cannot make a protein. First, DNA has to transcript an RNA, then a protein is formed. The protein synthesis machinery has evolved around RNA. The above discussion indicate that both RNA and DNA can function as genetic material, but DNA, because it was more stable, it means more stable, is preferred for storage of genetic information, right? For the transmission of genetic information, RNA is better. Transmission of genetic information, RNA is better. But storing of genetic information, DNA is better. Right? Now RNA world. Altman, after the discovery of Altman used this one RNA world, right? From foregoing discussion, an immediate question becomes evident that which is the first genetic material. So this RNA is the first genetic material. We know it shall be discussed in detail in the chapter on chemical evolution. Altman discovered this one. RNA is the first nucleic acid which is formed, right? But briefly, we shall highlight some of the fact and points. RNA was the first genetic material. First thing is there. There is no enough evidences that suggest that essential life process such as metabolism, translation, splicing, etc. they are present in RNA only. Evolved around RNA. RNA used to act as a genetic material as well as a catalyst. In some cases, which RNA is act as catalyst? It is in bacteria, in prokaryotes it is 23 sRNA, but in eukaryotes it is 28 sRNA. They act as catalyst or they are act as ribozyme. There are some important biochemical reactions in the living system that are catalyzed by the RNA catalyst and not by the protein enzymes. RNA catalyst means what? Ribozyme, through ribozymes, right? And which process? We can say protein synthesis only due to the ribozymes or RNA catalyst. But RNA being a catalyst was reactive and hence unstable. Therefore, DNA has evolved from RNA with chemical modifications. So, first RNA is formed, then due to some modification, some mutation, we can say mutation and modifications happens, chemical modifications happens. Chemical modifications is what? Nucleotide, second carbon has no, that means they lack oxygen. Now they become deoxyribose. Simply, thymine was in uracil. What happens? A methyl group attached with uracil and it becomes a thymine. So, some here modifications, chemical modifications that make it more stable. DNA being double stranded and having complementary strand further resist the change by evolving a process of repair, right? So, RNA world means which one is the first genetic material? It is RNA, right? Because RNA is independent. Replication. Now, how a replication performs? Replication means what? A DNA forms another DNA. So, it is a semi-conservative process. You can see here in this diagram, right? This is the parental DNA. This one. Now, this strand is separated. This is unzipping of DNA. Unzipping of DNA, but that means look here. This one is parent DNA, right? This one is parent DNA, and another this one. Only this one is daughter DNA strand. Means one is already parent DNA and daughter DNA. Half is already formed because it is a parents, and another this one daughter is newly formed, right? Because half strand is formed, so it is called semi-conservative. Same is half. Okay, that is replication. You understand why how a nucleosome is formed? You can see here. For the formation of a nucleosome, what happens? We know here histone protein, histone, octamer, and histone 1 monomer, right? And DNA. Helix. These three components, they are responsible for the formation of histone protein. And look here, this is a octamer. Octamer means 1, 2. It is like a relay. 3, 4. When you see this one is dimer, this one is also a dimer, histone dimer, right? These are histone dimer, and this one is called tetramer. Right? Now tetramer means it is made up of 4 units. This one is 2 units. This one is also made up of 2 units. That's why it is called dimer. Now total means 4 plus 2 plus 2 is 8. 2 dimer and 1 tetramer is here, right? Okay, now you see here, this one is a monomer. This one is monomer protein, right? Now when DNA is, suppose that this is the DNA. Now this DNA, it comes from that side and first circle, now second circle, but third circle is not completed. Now it comes to that side, right? And this goes to that side. This is that part. This one is called nucleosome. Do you understand? It contains 200 base pair. How many? 200 base pair. How 200 base pair? Here it contains this side. This side it contains near about 150 to 170 base pair, right? And remaining base pair is here. Near about 30 to 40 base pair. Here 40 base pairs are made up of this one. So this is a nucleosome body, right? Okay. Now when you see this one is coming here, right? Then what happens? It come here. Now here another aptamer is there. Look, this is the another aptamer. Okay, and here when it comes from that side, what happens? Here another dimer, tetramer. It comes from that side, one circle, then second circle, and then come to that side and goes to this. Now another comes, another one, monomer. Now aptamer is there, right? So do you understand here? One circle, two circle, right? And third circle is from this side. Just we crosswise here and come to this side. So when we see, we understand there that one is that side, that like structure is formed, and there is that side. That type of structure is formed, and this one structure is called chromatin, and these are called beads on a strand, right? This one is it is H1 histone, and this is a monomer. Okay, so this is the structure of nucleosome. Do you understand how a nucleosome is formed? And that is called packaging of DNA molecule. Okay. Replication. While proposing the double helical structure of DNA, Watson and Crick had immediately proposed a scheme for replication of DNA to create the original statement that is followed. It is the same statement which is given by Watson and Crick in his own word, right? We cannot change anything here. What is? It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. Watson and Crick in 1953. This is the direct statement which is said by the Watson and Crick. The scheme suggested that two strands would separate and act as template for the synthesis of new complementary strands. They act, or both strands, they act as template, right? After the completion of replication, each DNA molecule would have one parental and another newly synthesized strand, that is daughter strand, and this scheme was termed as semi-conservative DNA replication. As we see here, this here, right? Do you understand what is given? We can see here daughter, that is daughter DNA strand, DNA, not a DNA strand, it is a daughter strand, right? Daughter strand that is newly formed, we can say new, and that one is parental, because half is parental and half is newly strand, so it is called as conservative, semi-conservative, right? Semi-conservative, and both strand they act as template strand, right? So the experimental proof, how this experimental proof is given. It is now proven that DNA replicates semi-conservatively. It was shown first in Escherichia coli, E. coli, right? And subsequently in higher organisms such as plant and human cell also. Matthew Michelson and Franklin Stahel, simply we said them Michelson and Stahel, performed the following experiment in 1958. What experiment is there? They grew E. coli in a medium that contains ammonium chloride, but this ammonia is heavy ammonia, right? Heavy ammonia. Nitrogen N15 is the heavy isotope of nitrogen, but it is not a radioactive, right? As the only nitrogen source for many generations. Nitrogen, because nitrogen is essential for the formation of nucleic acid, nitrogen base there. The result was that amino N15 was incorporated into the newly synthesized DNA as well as other nitrogen containing compounds. This heavy DNA molecule could be distinguished from the normal DNA by centrifugation in cesium chloride, CsCl, density gradient, right? Because this N15, it is not a radioactive, it is given here. Please note that N15 is not a radioactive isotope and it can be separated, right, from N14 only based on densities. So we use this density centrifugation here, right? Means this, and from ammonium chloride, this heavy nitrogen is used in the formation of DNA. So that means the new DNA which is formed, it contains N15 only. Then they transfer the cell into the medium with the normal ammonium chloride. Now this normal ammonium chloride is the normal nitrogen, it is incorporated with the newly DNA, and took sample at various definite time intervals as the cell multiplied. And we know bacterial cell they multiply in 20 minutes. Means in case of E. coli, right, every various interval times, means after 20 minutes, then 40 minutes, then 60 minutes, then 80 minutes, he took samples, right, and extract the DNA that remained as double stranded bodies, right. The various samples were separated independently on CsCl gradients to measure the densities of the DNA. Densities of DNA, right? This test tube, you can see here test tube, right, and you can see here, because more density means they are located at the base, less density means they come to the upper portion, right, toward the surface, toward the upper region, not bottom. Heavy means toward the bottom, less heavy means it is in the middle, and if it is light, it is in the upper portion. This one is in middle, and light heavy means no heavy is there. Here in this, can you recall what the centrifugal forces is, and then think why a molecule with higher mass density would sediment faster? We can say, right, okay. As for example, centrifugation means what? Centrifugation force means what? Okay, you understand how we separate a butter from the curd. Simply it is due to the centrifugation, that process. The centrifugation, right? Okay, here. Separation of DNA by centrifugation is given. It contains only heavy DNA. Look here at the base. Gravitational force is more here. After 20 minutes, when two daughter cells are formed, so this DNA, parental DNA is there, one parental and one N14 DNA is there. N14 is given in green color and N15 is given in red color here. So generation first means is total hybrid. Both generation are hybrid, right? 100% is hybrid. Now in second generation, that is 40 minutes. After 40 minutes, what happens? This heavy is only 2. So this heavy means hybrid. They forms 2 hybrid. So one from this one is means when heavy DNA it forms a hybrid, and another is light DNA. It forms another light means what? It forms only a light. Second light DNA. Now we have a 4th generation means what is the percentage? Here we can say 50% are light and 50% are hybrid DNA. Okay. Sometime what happens? The different another type of questions will come. As for example, if it is given, look, 40 minutes means we have 50% heavy and 50% hybrid and 50% light. But then after 60 minutes, what happens? 60 minutes, what happens? These it forms 2, but both are light. It forms 2. One is light, another one is hybrid. It forms 2. One is hybrid and the form light. This form 2 means both forms light. So what is the percentage here? We understand only 2 is the parental. These two are hybrid. And 1, 2, 3, 4, 5, 6. 2 into 6. What is the ratio? 2 are hybrid and 6 are light. Right? Okay. In 80 minutes, what happens? 80 minutes of only 2 is heavy, and remaining is what? 1, 2, 3, 4, 5, 6, 7, 8. In 80 minutes, 16 equally is formed. In 16 equally, only 2 are hybrid and remaining 14, they are light. We can say here 2 into 8. What the ratio is? 2 into 16. 2 into 16 means what? 1 is to 8 ratio. Right? Here what is the ratio? Here it is 1 into 3 ratio. 1 is to 8 ratio means what? We can see. We have to calculate the percentage, right? 50%. Then what happens here? 25%. Then what happens here? 12.2%. Then what happens here? Less than 12.2 means what? It becomes 6.125%, right? Hybrid DNA. If you understand that, what he understand here? Here he understand that this one is parental and one other is newly formed. Means what it is? He said them semi-conservative. It proves that DNA replication is semi-conservative method. Thus, the DNA that was extracted from the culture one generation after transfer from N15 to 14 medium, that is after 20 minutes, because E. coli divides in 20 minutes, that had a hybrid or intermediate density. Hybrid means what? One strand is parental and the other one is new strand. DNA extract from the culture after another generation, that is after 40 minutes, so 2 generations, that means we can say 2 generations, was composed of equal amount of this hybrid DNA and equal amount of light DNA. If E. coli was allowed to grow for 80 minutes, then what would be the proportion of light and hybrid density of DNA molecule? So I tell you here, because only 2 DNA is hybrid, right? But 80 minutes means what happens? How many generations are going on? 4 generations, right? So first generation means 2, second generation is 4, third generation means 8, and fourth generation means 16. So we know 16, it is, what is the ratio? It is 16 is to 2, or we can say it is 8 is to 2. 8 means light and 2 means what? It is hybrid. This is the ratio in 80 minutes. 80 minutes means 4 generations, right? If E. coli was allowed to grow for 80 minutes, then what would be the proportion of light and hybrid density of DNA molecule? It is 8 light and 2 hybrid. Very similar experiment involving use of radioactive thymidine to detect distribution of newly synthesized DNA in the chromosome was performed by Vicia faba. It is the scientific name of faba beans, right? And this experiment was given by Taylor and his colleagues in 1958, right? and the experiment proved that DNA in the chromosomes also replicate semi-conservatively. Right? Now it is clear that DNA replicates semi-conservatively. One strand is parental and the newly strand is formed. Okay. Now in next class we will read the machinery and the enzymes for DNA replication. Okay. So today we discuss here only by NCERT. Read this NCERT very carefully because all the questions in NEET exam only NCERT book questions will come. Okay. Thanks.